Welcome to Conversations in Science. My name is Gabrielle Graham, and I am the Public Programs and Community Partnerships Manager at the Buffalo Museum of Science. The goal of these programs is to encourage scientists and researchers with the opportunity to informally chat with the community about their work and trending science topics. These conversations are designed to be engaging for science-interested individuals, but also to help scientists improve their communication skills. Communicating research outside of a scientist's area of study is an important skill, often requested in grants and education initiatives. So public forums like this one help our scientists as much as they help the people who are gaining a little bit of new knowledge. We like to thank all of our audiences for their continued support of our educational programs. The support of our membership enables us to continue our mission and to serve our community. So now let me introduce our speaker. This is Steven Fleck. Steven is a PhD candidate in Dr. Victor Albert's lab in the Department of Biological Sciences at the University of Buffalo. The Albert Lab conducts research with genome evolution and population genetics in plant species. Today, he'll be speaking with us about a project to sequence ash trees, something quite different from their usual plant research. And with that, I will turn it over to Steven Fleck.
Hi, thank you. Let me just pop my slides up and then we'll be all set. Okay, sure. Okay, so as Gabrielle said, let me just turn the brightness down. Okay, so as Gabrielle said, my name is Steven Fleck. I'm a PhD candidate in Dr. Victor Albert's lab at the University of Buffalo. And today I will be giving a talk titled Conversations in Science, a Dash for the Ash, Critically Endangered Ash Trees. So one thing that I want to say up front is that I tried to assume that a lot of the audience wouldn't have the same background that I have. So I'm going to try to slow things down. I'm also going to try to explain things, maybe go through a couple methodologies and why we can make certain claims that we make. And hopefully everyone's able to follow, but again, feel free to ask any questions. Sometimes I may mention something and then explain it shortly after. So sometimes you only have to wait a moment or two to get it. Okay, so to begin with, I just want to introduce my lab and the co-authors of this study. So in the middle is Dr. Victor Albert, and the other two yellow arrows point to Michaela Richter, a master's student, and me, a PhD candidate. So on the left-hand side, you have Dr. Charlotta Linkvist and her two PhD candidates, Flavio Coelho and Crystal Tomlin. This project is a combined effort between the two labs. We did the sequencing and the collection of the species together. So the Albert Lab primarily studies plant genome evolution and has worked on several well-known plants, including gardenia, coffee, the $5 tree in Asia, syzygium, which is the world's largest tree genus, as well as carnivorous plants such as Utricularia and Pinguicula, just to name a few. So I also want to mention that our lab doesn't typically work with ash trees or really any plants native to our area. And this fact became important because after sequencing and assembling the genomes of our 3 critically endangered ash trees, a chromosome level reference assembly for one of our species was published by Huff et al. In late 2021. Now, if you don't know what a reference genome is, it is a high quality genome assembly that can be representative of a group of related species. So ash trees typically have 23 pairs of chromosomes, similar to humans. So this reference would also have 23 chromosome size sequences in it as well. So one of the things that you can do with a reference assembly is create something called a reference guided genome assembly for other species that are closely related, and they actually did this as well. So they took some existing lower quality Ash genome assemblies that were created using short read sequencing, which I will cover shortly, and stitched them together using the reference genome as a guide to create chromosome level assemblies for our other 2 critically endangered ash trees as well. It's not all bad news though, because now that we have this reference available to us, we can use it to our advantage for stitching together our own long read assemblies, compare that to the short read assemblies I mentioned before, into our own chromosome level pseudo assemblies. And then we can make comparisons against their reference guided genome assembly, or sorry, the reference guided chromosome level short read assemblies. But that will be a little bit later on in the presentation. So we had the opportunity to dabble into the world of ash tree genomes through the org.1 project, which is currently in its pilot phase. The goal of org.1 is to support faster, more localized sequencing of critically endangered species by enabling biologists to rapidly sequence those species close to the sample's origin using the latest ultra long read approaches. So they provide us with free of charge consumables on the condition that the data will be publicly available on the European Bioinformatics Institute, or EBI, public database. So org.1 is supported by Oxford Nanopore Technologies, one of the leaders in next generation sequencing, which is great because our lab has access to one of Oxford Nanopore's GridIONs to sequence our tree genomes with. And I'll actually show you guys what a GridION sequencing platform looks like on the next slide. So, oh, we'll admit someone.
Yeah,
okay.
I'll take care of that. Don't worry.
Okay, sorry. Okay, so now I want to mention the overall purpose of the research. And like I said, on the right-hand side here, I have the GridION, and this is a flow cell that you'll hear me talk about, a single flow cell, and the arrow points to where we put it into the GridION. So here we demonstrate that data from a single Oxford Nanopore GridION cell can, even in the absence of highly accurate short DNA read polishing, produce highly accurate de novo genome assemblies. Those are genome assemblies that are created without a reference, that are adequate for downstream analyses, such as synteny evaluations, which compares 2 sequences within or between species.
You've frozen a little bit. I think I was waiting to see what happened after synteny associations. So we are, oh, here we are. We lost you a little bit. So if you could just pick up again from the synteny analyses.
Okay, yeah, thank you. Sorry that I cut out. So we can do synteny evaluations, and this compares 2 sequences within or between species that have conserved gene order. We could do ploidy evaluations, which explore the number of whole genome duplications that an individual experienced in their long evolutionary history. We could do paleodemographic analyses, which look at effective population sizes over time, and phylogenomics, which show how different individuals and species are related to one another. So next, I will briefly cover the Oxford Nanopore long read sequencing and why we used it for this project.
Quick, before you head into the sequencing there, this is a really basic question. Why do we need, or why do we want to sequence a genome? Like, I know it's good to have knowledge, and it is important to understand the world around us, but what types of information are you getting out of comparing genomic sequences? So, I mean, I think I heard you say evolution. You can think about the ways that species have changed over time. When you mentioned paleodemographic analysis, kind of what has remained the same and what has changed in the age of, or, you know, between different individuals in the line. Is that accurate?
Yeah, so the thing with when you do whole genome sequencing and creating these genome assemblies so previously some of the work that we've done has only focused on, I say we, but just in the general biology community, has focused on specific genes. So they weren't looking at a whole genome approach, but part of that was due to the cost. Part of that was due to just technology not being there yet. And a lot of the things that I talk about, like when I'm talking about whole genome duplications, when I'm looking at things like, population size history, those are things that sort of require whole genome data to really get like a complete picture of those type of evolutionary events. And they really help us understand those events in evolution a little bit better as well. Just having access to those full genome sequences kind of gives us, plenty to work with.
Excellent, thank you.
Okay. Okay, so so I said I was going to talk about the grid ion, but this is actually a diagram for a different Oxford nanopore product. This is the min ion, but the premise is exactly the same. So after we isolate the DNA from the plant's leaves, those DNA libraries are loaded onto a port where it then flows onto a membrane with a couple thousand nanopores embedded into it. And I actually have that little, that plane of nanopores marked with an arrow on the screen. So as DNA is translocated through a nanopore, the grid ion measures a change in electrical current, and an ever improving base calling algorithm translates that into DNA reads for us to use. Even though it keeps improving, long read sequencing, it has relatively high error rates. And for our reads, it was about 10%. So that's a disadvantage, but one of the major advantages of this type of long read sequencing is that individual reads can be hundreds of thousands of base pairs in length, which that spans multiple genes, multiple repetitive elements and regulatory elements. And if we compare this to the highly accurate Illumina short read sequencing, which is an older sequencing technology that is still used today for its low error rates, and for it, honestly, it's a relatively cheap technology to use. These reads are typically between 150 and 300 base pairs in length, and that's very unlikely to span multiple genes. So if you're interested in synteny analysis, like I brought up before, which is dependent on conserved gene order or knowing what that conserved gene order is, you'll want to sequence reads that span multiple genes. Additionally, if you're looking at tandemly duplicated genes or genes that are duplicated one right next to each other, long read sequencing is much more likely to capture that tandem array compared with short read sequencing, which is more likely to collapse that array into fewer genes than are actually there in the genome. So now I want to focus on our species in the genus Fraxinus. So ash trees are dominant or co-dominant in many plant communities throughout Asia, Europe, and North America, but we're really only going to be focusing on species that can be found locally here in New York. So in 2017, all 5 species of ash that can be found in New York were added to the International Union for Conservation of Nature, or IUCN Red List, as critically endangered. With help from the Western New York Land Conservancy, our lab was able to collect sequencing material and vouchers from 3 species at sites in Brockport and Dunkirk, New York, right here. And that's about an hour southwest of Buffalo. So highlighted in yellow are the 3 species that we sequenced, which is Fraxinus americana, known as the white ash, Fraxinus nigra, known as the black ash, and Fraxinus pennsylvanica, which is known as the green ash. And the reference assembly that I mentioned earlier from Huff et al. Is actually also of the green ash, Fraxinus pennsylvanica. So previously, ash trees were very abundant, but in the last 15 years or so have become difficult to find healthy adults. And if you're like me and identify trees while you're driving, which, by the way, I do not recommend anyone here do that, but ash trees have become a lot easier to identify from a distance now, at least in the growing season. Unfortunately, it's because they're essentially the only standing dead trees on the side of the highway mixed in with the leaved out ones.
Oh, that's
Yeah, it's kind of a sad reason that we could identify things easier now. So the reason for this sudden decline of these ash trees is due to the emerald ash borer. And this is an East Asian beetle that was first discovered in Michigan in the summer of 2002. And by 2018 spread to 35 US states and 5 Canadian provinces. So while the adults cause little damage feeding on the leaves, the larvae actually feed on the inner bark, and that disrupts the flow of water and nutrients, essentially girdling and killing the tree. Infected trees have a high mortality rate, and since its detection, the emerald ash borer has killed hundreds of millions of ash trees in North America.
And girdling is, can you explain what that is?
Yeah, so girdling is actually a process of killing trees, and so people will also do it too. Like, for example, I was working for Ecology and Environment in about 2011, and any infected tree we actually girdled, which means you basically you have this transport tissue that transports like sugars down from the leaves and water up from the roots, and basically you kind of cut that off all the way around the tree, and that will end up killing the tree because you can't get water up and you can't get sugars down.
It's like effectively sawing the tree in half by just putting in not something as intense as actually sawing it in half, but
right.
Cutting a swath of the bark and the underlayer so that it can't transport up and down. Okay.
Yep. And girdling trees was a strategy that people used early on, but that's sort of outside my expertise. It was just something that I did a little bit while I was there. Of course, that didn't exactly stop things. So
yeah.
So yeah.
If you're interested in girdling, go talk to your arborist, not to your genomic scientist.
Right. Okay, so now on to a little bit about the emerald ash borer resistance in ashes. So other research groups have identified some natural resistance in some ash trees. Of course, some of these are present in the native range of the emerald ash borer. So along the x-axis of this plot are different species of ash, and on the y-axis are the proportion of emerald ash borer larvae that were killed by the host tree's chemical defense response. So if you look at the red, blue, and green highlighted areas, these are the species that we sequenced. And then I also have the gray highlighted species, and those are the other 2 New York species that we didn't sequence. So you can see that they're kind of on the lower end of the proportion of emerald ash borer larvae killed. And with these low levels of resistance, genetic diversity is quickly declining with the populations of our 3 species being wiped out across North America. So by comparing the genomes of resistant species, 53 candidate genes have been found to have convergent amino acid evolution. And this isn't work that I've done, this is work of other researchers. This means that so what it means to have convergent amino acid evolution, this means that the gene products between species are similar, but not because they share a common ancestor, but due to evolution after these species split from one another. So they've converged on similar sequences, despite those sequences not being similar prior to that.
Yeah, so just as an analogy on that, it's like bats have wings and birds have wings, but they do not have wings for the same reason. So we're looking at having a similar characteristic that comes from different evolutionary measures.
Yep. Yeah, and they looked into some of the potential roles that these candidate genes have. They found 7 that have roles in a biosynthesis pathway that have roles in herbivore and pathogen defense, and 17 others that relate to herbivore recognition, defense signaling, and programmed cell death. And they also found that susceptible species frequently have a loss of function mutation in these genes, which if I haven't looked, but if I had to guess, I would hypothesize that maybe ours probably have some loss of function mutations, considering that we have very low resistance to the emerald ash borer in the species we are looking at. So before I go into our genome assemblies, I just want to mention a few things about what a genome assembly is. So like I put on the slide, this is really nuclear genome assembly 101. So I will be showing de novo genome assemblies. I mentioned this before, but just as a reminder, these are genome assemblies that were created without a reference to help it along. And when you're working with a non-model species, like essentially all the species that we work with, there are typically no high quality assemblies from close relatives to help in the assembly process. So next, when I talk about—
Sorry, I'm just going to interrupt one more time. Not just one more time, many more times. The de novo assembly, if you were sequencing with one that did have a scaffold available, it just means that you have a genome of the same species that you're comparing the new genetic information to. Is that right?
So the reference, so when you're doing, there's de novo assemblies on one side that has no reference. It's just an assembly that's done from the ground up. And then you have a reference guided genome assembly, and that uses a particular genome assembly, even if it's not the same species. Yeah, it doesn't have to be the same species. It can just be a close relative.
Okay.
And that's actually part of the goal of the reference genome that was published in late 2021, so that they could use it as a reference assembly to do reference guided assemblies with.
Excellent.
Yeah.
Thank you.
Yep. Okay. So let's see. Okay. So another thing that I want to mention is when I talk about assembling a genome, for the most part, what I mean is that I'm assembling what's called a haploid genome. And what I mean by that is that inside every single one of our cells, humans have two sets of chromosomes. We have one set of 23 from each biological parent. This is probably where the company 23andMe gets its name from. So in a way, every individual person is a population of two, because in each one of our cells, we have two sets of chromosomes that are just a little bit different from each other. So conveniently, many ash trees actually have the same number of chromosomes as humans, 23. So it's sort of chromosome pairs, I should say. We have 23, so it's sort of easy to remember. So every ash cell that we ground up and extracted and purified DNA from started out as having 23 complete pairs of chromosomes. And that grinding, extraction, and purification process fragmented the chromosomes into smaller pieces. And then it was my job to piece those fragments back together into those 23 chromosome size pieces. But that would be a best case scenario. And many assemblies are not actually chromosome level, but they're still very useful for biologists. Lastly, the reason why the goal is 23 chromosomes instead of 46 is because the DNA sequences of a chromosome pair are very similar, and it is very difficult to know if any given DNA sequence belongs to one chromosome pair or the other. So one issue with haploid genome assembly is that the more different the chromosome pairs are from one another, this is also known as heterozygosity, the less likely we'll be able to reduce that pair of chromosomes into a single chromosome during the assembly process. So if a genome has high heterozygosity, you will assemble a haploid genome plus the parts of the other chromosome pair that were just different enough to not match perfectly. So that basically makes most assemblies partly diploid to begin with, at least. But this is okay because there are strategies for reducing partly diploid assemblies down to a haploid assembly. This can require a lot of sequence data, and we're only working with a single GridION flow cell for each species, which makes sequencing the ASH genome much cheaper and accessible, but it does limit the amount of sequence data that we have to work to solve these issues. So the last thing I kind of want to mention before I kind of get into some of our results is this other vital step of assembling a genome. And this is something called a genome annotation. So our assemblies were annotated using something called a program called the Gene Model Mapper, otherwise known as GEMOMA. This is a homology-based gene prediction software. And I'll explain what that means. So a genome assembly, or sorry, a genome annotation essentially gives us information about the gene content of an assembly. So it'll locate DNA sequences of a gene. It'll predict the RNA sequences, the RNA sequence that the DNA is transcribed into, and it'll predict the protein sequences that the RNA is translated into. So if you're unfamiliar with the central dogma of biology, it's actually exactly what I just mentioned. It's the process by which instructions in DNA are converted into a functional product. And actually, both RNA and protein are both functional products of genes. So GEMOMA annotates a genome using another species genome annotation. A close relative will likely give a better result than a distant relative. And it does this by identifying homology, or similar DNA sequences, between the two assemblies, and then transferring those annotations from the reference to the target genome. So GEMOMA is really useful because it means that we don't need to do any additional RNA sequencing in order to annotate our assemblies. We can use what's already publicly available to get the job done. And it also means that I don't need to cover how we would use RNA sequencing to annotate our assemblies. So I'm kind of thankful for that. So in our case, we used 4 species to annotate our 3 Fraxinus assemblies. We used Fraxinus excelsior, which is the European ash. We used olive, we used jasmine, and we used a well-studied but distant relative, a mustard known as the thale cress, Arabidopsis thaliana, which maybe some people might recognize the name Arabidopsis. So let's actually take a look at the initial assemblies now. Okay, so they weren't really too bad. On the far right-hand side, I included the reference assembly by Huff et al. And it serves as a good benchmark to compare our assemblies with. So if you look at the green highlighted cells, our 3 Fraxinus assemblies had comparable levels of completeness compared with the reference assembly. So this is measured using something called benchmarking universal single copy orthologs, or BUSCOs, which is a set of single copy genes that should be highly conserved among a group of related species. And us finding about 95% is a pretty good value to get. So still, our assemblies could improve. The assembly length for Fraxinus americana and Fraxinus pennsylvanica were a bit larger than estimated. And they have a higher percentage of complete duplicated BUSCOs. That's the value, the yellow highlighted value near the bottom. They have more than what the reference has. And this suggests that we just have too many duplicated genes there, meaning that this is another reason to think that we're dealing with partly diploid assemblies. So, like I said, this happens when you're trying to assemble highly heterozygous individuals. And actually, the highly haploid assembly of Fraxinus nigra might actually indicate or suggest that this individual was from a more inbred or homozygous population. Click. There we go. Sorry. And this is basically what we saw when we looked at the effective population size histories between our 3 species. So we can actually infer population size history of each ash species using what's called the PSMC model. So within every diploid organism, there are thousands of allele pairs. And each allele pair has an independent time since the most recent common ancestor, or Temerca. So even though we're only looking at a single individual, because we're all a population of 2, it's possible to gain interesting information about their ancestral population size histories and coalescence times. And we can see this because at one point, all 3 of these species belonged to the same population. So at some point, these curves should meet together and have a similar pattern. Now, you may notice that Fraxinus nigra in green never actually connects with the other species. And this may be due to it being more inbred than the other species. A recent study showed that simulated selfing or reproduction with itself resulted in PSMC plots that were shifted towards smaller population sizes and more recent coalescence times. So on the left, the pink curve represents the population size history of the original individual. The pink curve represents the individual after one generation of selfing, and the blue curve represents an individual after 2 generations of selfing. So you can see that this almost looks very close to what I ended up seeing with my Fraxinus species, Fraxinus nigra specifically. So what I kind of wanted you to take away from the slide is that our Fraxinus nigra likely came from a population with lower effective population sizes than Fraxinus americana and Fraxinus pennsylvanica. And lower population sizes correlate with lower heterozygosity within a population and a decreased ability to adapt to environmental stresses, because evolution can only work using the standing variation. And if you have very little variation within the population, there's not a lot for evolution to work with. Okay, so next, I want to briefly focus on these KS density plots. And I'm going to kind of take a minute or more to kind of explain how these are generated and what they mean. So these are created by mapping all the genes within a genome against themselves. So, or basically every gene against every other gene within the same genome. And here we're only looking at syntenic gene pairs or gene copies that also share neighboring gene order. So the reason that we're only using syntenic genes here is because if they share neighboring gene order, you can assume that they came from a single ancestral sequence and resulted from a larger scale duplication event. So if you create frequency plots for synonymous substitution rates between those syntenic gene pairs, you'll end up with these types of plots. And I'll just explain synonymous substitutions really quick. So because we're working with synonymous substitutions, these are mutations within a gene that do not change the protein that that gene codes for. So these should accumulate within a gene like a ticking clock. And the more similar a syntenic gene pair is, the more recent their duplication event is that created it. And the more divergent the syntenic gene pair is, the older the event is that created it. So on these plots, you'll see large spikes that represent a bunch of genes doubling at once. This would be representative of a whole genome duplication or triplication event. And we'll talk about the importance of those a little bit later. So actually, if we look a little closer to the left side of the plot, there is this small hump that can be seen mostly in Fraxinus americana and Fraxinus pennsylvanica. So these are syntenic genes that are very, very recent and very, very similar. And these are actually the diploid parts of the genome assemblies. So they represent the current differences between the chromosome pairs, essentially just different alleles of the same gene or heterozygosity. So heterozygosity is also evident from a bimodal depth of coverage peak, but I'm not going to cover that in this presentation, but I will just show you what it looks like when you get rid of those sequences. So using a tool called purge haplotigs, I reduced the diploid assemblies down to haploid assemblies, and you could actually see that with all 3 assemblies now having a single haploid peak. We re-annotated the assemblies and recreated the KS plots, and then you could see that the diploid sequences are mostly gone. Okay, so here are the final haploid assemblies. We won't be talking about genome stats anymore after this. Thank goodness, I'm sure some of you are thinking. But each assembly is now much more contiguous, and the BUSCO scores didn't decrease much. In fact, after purging the heterozygosity, BUSCO actually, we identified more BUSCOs in both Fraxinus americana and Fraxinus nigra. The genome sizes were much more close to the estimates that were expected, and the complete duplicated BUSCOs are much closer to the reference assemblies. So now we have good haploid assemblies to work with. So next, I want to look at the ploidy level, or number of whole genome duplications that each Fraxinus experienced in their evolutionary histories. So in the bottom right, I have a dated phylogeny. So basically, a dated phylogeny just kind of shows relationships between species, and it kind of dates, it gives a timing for when these species branched off from one another. So this shows the expected polyploidy events for Fraxinus. We have a star in the bottom right that represents a recent whole genome duplication. And then we have two triangles, a green one and a red one, that represent two more ancient whole genome triplications. So the oldest, the red triangle, the oldest one, actually represents what's called the gamma triplication, and this is present in all core eudicots. And core eudicots make up about 70% of all flowering plants, everything from roses to daisies and all their relatives. So also, the gamma triplication is not found in basal eudicots, which include barberries and buttercups, if people are familiar with those. So we already showed the KS density plots with the two peaks for the polyploidy events, but now I kind of have them approximated on the density plot now. Unfortunately, these old events like gamma, like the gamma triplication event, they're pretty difficult to see in species with subsequent polyploidy events, but we do know they're there from other analyses. So I want to compare my assemblies against the reference Fraxinus pennsylvanica assembly, the chromosome level one. And when we look at that comparison, we should expect to see one KS peak for the recent species split, and then two KS peaks for their shared whole genome duplication events. I also compared the assemblies against grape. I just highlighted where grape is on the time tree. And grape is actually a really good benchmark for comparing core eudicots to, because grape hasn't experienced any additional polyploidy events since the gamma triplication in the ancestor of all core eudicots, and it also has a relatively low mutation rate. Okay, so for the first comparison, we see the predicted KS peaks on the right. I have them marked on the plot. And we have the recent species split, and we have the two shared polyploidy events. So as I mentioned earlier, the older gamma triplication event is just a little bit too old to see here. But we also see evidence that both species have undergone a recent whole genome duplication on the syntenic dot plot on the left. So on the left-hand side, we have this syntenic dot plot. So what this is on the x-axis, we have the Fraxinus pennsylvanica reference assembly, and you can see it has 23 chromosomes. And then on the right-hand side, we have our Fraxinus americana assembly. Everywhere where there's a dot represents a syntenic gene pair, and the color of that dot correlates to the colors on the KS plot. So, so the purple ones, those ones, you can actually think of those as being red and blue. They represent the red and blue, and then the green dots represent that older whole genome triplication that happened about 80 million years ago.
So is it usual to, or the fact that the red and the blue are mainly co-occurring in all of these spaces, is that something that's seen through other sequencing, or is this something that was unusual in this result?
No, this is what was expected, but also the purple, unfortunately, the purple, if it was a perfect, if I perfectly purged the heterozygosity, the purple wouldn't be there. And the blue and red, they would be somewhat overlapping, because those are basically the most recent sequences that they shared together. So so it makes sense that they would be kind of overlapping.
Oh, I see. Yep. Mm-hmm.
Yeah. Okay. So so on the plot on the left hand side, you can actually you can make out the most recent polyploidy event, because they'll look like really nice lines, like really nice purple lines. And the older polyploidy event that's made up of the green dots is much more fragmented, but this is because over time, genomes, gene copies will build up mutations and translocations. So that's why the green dots aren't nice lines like the purple lines are. So, click here, there we go. So if we start at the bottom, which is the chromosome level Fraxinus pennsylvanica assembly, and draw a line straight upwards, for every one gene in Fraxinus pennsylvanica, there are 2 syntenic blocks in Fraxinus americana. So you could see that 1, 2, but then if you start on the left-hand side, which is the Fraxinus americana the Fraxinus americana assembly, and draw a line directly over, you'll see that there's also 2 syntenic blocks of genes for every one gene. So this is a 2-to-2 ratio, which is what we would expect because we believed that they shared that whole genome duplication event in their recent history. And actually, we see the same patterns with Fraxinus nigra against the reference assembly. And we see the same pattern with our Fraxinus pennsylvanica assembly against the reference. Now, before I show the next set of dot plots between our species and grape, I want to briefly focus on the older whole genome triplication event that I have marked on the plot glowing in yellow there. So this is the Oleacy triplication event. The Oleacy is also known as the olive family, and this includes ash trees, olives, jasmine, and other popular ornamental plants like privet, forsythia, and lilac. And it was discovered by Zhu et al. In 2021 and occurred about 50 million years ago. Before last year, it was thought that multiple whole genome duplications could describe the pattern that we were seeing in the Oleacy. But using collinearity analysis, these researchers found that homologous gene pairs between grape, jasmine, and olive that suggested that a whole genome triplication occurred for all the Oleaceae, followed by a whole genome duplication in olive's evolutionary history after it split with jasmine. So if you look at the highlighted genomes on the figure on the right-hand side, one grape chromosome maps onto 3 jasmine chromosomes, and each of those jasmine chromosomes maps onto 2 Olea chromosomes. And actually, it would be the case that if we had Fraxinus there instead of Olea fragrans, it would also map to the Fraxinus assemblies as well. So one of the reasons that whole genome duplications are interesting is because these same researchers found out that stress-related and flowering-related genes were located in these conserved triplicated regions. And this suggests that polyploidy events may have contributed to the adaptive potential of the Oleaceae. So previously, it was hypothesized that while polyploidy events don't add variation to the genome, they can add gene redundancy for selection and genetic drift to act on. So now let's look to see if the triplication, let's see if we can see the triplication in the syntenic dot plots between our ash trees and grape. Now I'll just say now that we should expect a 6-to-1 gene ratio, because after Fraxinus diverged from grape, it experienced a whole genome triplication, just like jasmine, followed by a whole genome duplication, just like olive. Okay. So here's the syntenic dot plot with Fraxinus americana on the y-axis and grape on the x-axis this time. So again, each dot plot represents a gene with conserved neighboring gene order in both assemblies. So it's likely that they share a common ancestral sequence that was duplicated in a large scale duplication event. So as mentioned, when we compare the syntenic dot plots against our assemblies in grape, we should be seeing the 6-to-1 gene ratio. And essentially what this is one gene tripled into 3, and those 3 doubled into 6. and I have a zoomed in section of the dot plot on the right-hand side. And if we draw a line straight upwards from the bottom, we should roughly see a 6:1 gene ratio. And I drew boxes around that to make it a little bit easier to see. So you can see that some of these have a fewer than a 6:1 gene ratio. And that's because after a gene duplication event, the most common fate for one of the two gene copies is non-functionalization. So it'll build up so many mutations that it will be no longer recognizable as a gene pair. So we actually see, again, we see the same relationship with Fraxinus nigra against grape. And we see the same with Fraxinus pennsylvanica against grape. And of course, we actually would also see this with the reference Fraxinus pennsylvanica against grape. So the dot plots are informative, but we can actually get additional evidence for the whole genome triplication using something called a fractionation bias analysis. And because we had that reference genome assembly, we ended up creating our own chromosome level pseudo assemblies for our species by stitching together our assembled contigs using the Fraxinus pennsylvanica reference as a guide. So this allows us to look at longer stretches of genes for comparative analysis against other ash species, jasmine, olive, grape, or really whatever else we wanted to compare it to. So on the x-axis of a fractionation bias plot, we have windows of genes for a target along the chromosome. And on the y-axis, we have the percentage of gene retention following a polyploidy event. So essentially, after a species split and subsequent polyploidy events, a fractionation bias plot will show us the number of polyploidy events that the query species experienced relative to the target species. It can also suggest whether the polyploidy event was created through an in-species genome doubling or doubling through hybridization. And I'll kind of touch on that in the next slide. So I'm only really going to show a couple examples here to get this point across. But on the left, we have, we mapped jasmine onto grape. And we saw basically exactly what we would expect. We see a 3:1 ratio. And this is because jasmine and all the other Oleacy have that whole genome triplication since it diverged with grape. But again, unlike olive and ash trees, jasmine only has this whole genome triplication and no other polyploidy events. So the reason that we see two jasmine chromosomes with similar patterns of gene retention in blue and green, and then one that's a little bit more divergent in red, this is because this triplication event likely occurred because of a hybridization event. So two chromosomes are from one ancestor and are favored in terms of gene retention, and a single chromosome is from another ancestor, which isn't favored in terms of gene retention. So there is a, that's why it's called a fractionation bias plot, because we can see a bias in gene retention for that one chromosome compared to the other two. And this pattern is actually common among polyploidy events that occur due to hybridization, regardless of whether they're whole genome duplications or triplications. Now on the right-hand side, when we map an ash tree against grape, we see a 6:1 ratio. More specifically, we actually see 3 groups of chromosome pairs that have similar percent retention patterns. And this is, again, I'm being very repetitive here, one chromosome tripled into 3 in the ancestor of the olive family. And then those 3 doubled into 6, which would make sense that we have three groups of chromosomes, 3 groups of 2 chromosomes that both Fraxinus and Olea have. So we can also compare ash to jasmine directly just to make sure that this pattern holds up. So when you map ash onto jasmine, you should see 2 ash chromosomes mapping onto 1 jasmine chromosome. So here we actually see on the left-hand side, we have Fraxinus americana chromosome 6 and 15 mapping onto a single jasmine chromosome, chromosome 12. And then when we map jasmine onto Fraxinus americana, we see that chromosome 12 maps onto both chromosomes 6 and 15. So it is a true 2:1 gene ratio relationship. Okay, so I'm only showing a couple examples here, but it does hold up for all species comparisons, and it's also worth mentioning why there are so many small chromosome pieces with low gene retention, kind of crowded at the bottom, and this is because these are sequences from older events, such as the triplication event, and even the Oleacy triplication or the gamma triplication. I said that the syntenic gene ratio between Fractionation and Jasmine was 2 to 1, but it's more like 18 to 9 if we count gene duplication starting at the gamma triplication. So 1 tripled to 3, that's gamma, 3 to 9, that's in the Oleacy, and then 9 to 18, that's for Fractionation and Olea. So there are older chromosome copies that have just had more time to diverge from chromosome 12, for example, of Jasmine. So they match, but they don't match that well. So here's a species tree generated from the protein sequences, summarizing the polyploidy events. So for simplicity's sake, I only highlighted the polyploidy events for grape and the members of the Oleaceae. So all species on the tree have the gamma triplication in the top left, and then the Oleaceae has another triplication at the bottom in red, and then olive and ash have an additional whole genome duplication in blue. So one last thing I wanted to focus on is comparing our long read chromosome level pseudoassemblies that were used for our fractionation bias analysis to the reference guided assemblies created by Huff et al. So you can see here that the self-self syntenic dot plots look almost identical, which is probably a good sign. And our genomes had nearly identical BUSCO scores, about 95%, so they were pretty complete and accurate. In terms of the genome annotations, we found roughly the same number of genes, around 37,000, but our genome annotations were more complete. So you can also assess the annotation completeness using BUSCOs, and our annotation outperformed there. So we found about 95% complete BUSCOs, well, they only found about 88. So, so our annotations were a little bit better. Now, when we compare the self-syntenic dot plot for Fraxinus nigra, you can see that more of our assembly was able to be stitched together using the reference assembly. And again, these genomes were of similar completeness. And again, our annotation outperformed theirs by a similar BUSCO margin. So one of the differences between our genomes was the number of tandem duplicated sequences that were captured in our assembly, but were missed in theirs. So as I mentioned before, if you're interested in conserved gene order or tandemly duplicated genes, you'll want to use long read sequencing. And this is because single reads span multiple genes and won't collapse them into fewer duplications like short read sequencing might. And that's because they don't span multiple genes, like I had said. So when we just simply look at the number of tandemly duplicated sequences between our assemblies and theirs, ours being the blue line and theirs being gray, you'll see that we both have a higher number of tandems and a higher frequency of those tandem arrays in almost every case. So you can also look directly at the genome and do a one-to-one comparison of the genes and see if their genome is actually missing the tandemly duplicated genes that ours has. And you can actually compare the exact same chromosome of our assembly to that same chromosome of their assembly. And I'll kind of show you what that looks like now. So if we compare, okay, so this is a comparison. So I'm looking at a group of tandemly duplicated genes that are surrounded by syntenic unduplicated genes between these two chromosomes. So the top genome is our genome, and on the bottom is that short read genome assembly from Huff et al. So the small green and gray rectangles are actually gene models, and the red lines are connecting similar sequences between the two chromosomes. So if we just focus on the tandemly duplicated genes, we can see that our genome assembly has several tandem gene models that aren't included in Huff et al.'s assembly. And I marked those with a large black bracket. So when we add in the red connectors, we see that there are indeed tandemly duplicated sequences that were missed by the short read assembly. And just for scale, the bracket that I added covers about 600,000 bases, which is still shorter than the longest nanopore read that we use to assemble this genome. As a reminder, their assembly was made using short reads, and those are only 150 to 300 base pairs in length, rather than this segment that I have here, that's 600,000 base pairs in length. So I chose this specific gene to look at the tandem duplications, not for any specific function that it had. I was simply making a comparison between the tandemly duplicated sequences between our long read genome guided assembly and Huff et al.'s short read reference guided assembly. It would be more interesting to look if any of those 53 candidate genes for emerald ash borer resistance were duplicated in our assembly and not theirs. Unfortunately, our 3 species do lack emerald ash borer resistance, but tandems could be being missed in species with resistance that we didn't sequence. Okay, so moving forward, it would be interesting to see if any of the candidate genes were in conserved triplicated or more recently duplicated regions, similar to the analysis that was that discovered the stress-related and flowering-related genes conserved in triplicated regions of the Jasmine genome. It would also be interesting to see if any candidate genes were tandemly duplicated but missed by Huff et al. I also haven't taken a close look at the gene models of the tandems that I showed on the previous slide. And these really do need to be investigated further to see if they are likely functional or non-functional gene copies. So in conclusion, it's possible to generate high quality genome assemblies with single GridION flow cells. The read coverage was sufficient to eliminate the diploid sequences that the long read assemblers struggled to collapse. Our assemblies were, they had comparable levels of BUSCO completeness with the reference assemblies, and our annotation had higher levels of BUSCO completeness than theirs. We also identified tandems that were missing from, we gave an example of tandems that were missing from Huff et al.'s short read reference guided assembly. And all 3 species were sufficient to support the newly proposed Oleacy triplication, as well as the subsequent whole genome duplication in both Olea and Fraxinus. And while Fraxinus was the group we chose to work with for this project, it's more of a proof of concept that a similar analysis could be capable with other threatened and endangered species before these genomes and insights into evolution are lost forever. So thank you, and I'll take any questions that anyone has.
Excellent. Thank you so much. So I will admit that I am a little over my depth in a lot of this, but I really appreciate being able to see, I mean, so I come from a naturalist tradition, you know, citizen science and identifying things on site. And so I really appreciate the amount of work that can go into identifying species characteristics through single cells. So you said the material that's needed for conducting the flow analysis is it's not anything that it just seems readily accessible. You know, when I'm thinking about from the museum perspective, anytime that you test something, you're destroying the object a little bit.
Right.
And so in this, there's very little destruction that goes on, a very small amount of material that can be used. I think that's just a fascinating aspect of this as well.
Yeah, so just to make sure we're clear, we did grind up full leaves for this process. And we actually took as much material as we could and preserved it because you know, these trees were already difficult enough to identify in their current state, which I think maybe all the trees that we collected from were in one, I shouldn't say all the trees, some of the trees that we collected from were in one state of degradation due to the emerald ash borer. So there was a chance that, let's say if we waited to collect the material next year or this season, those trees might not have really been there for us to identify and to collect the material from. So we did collect a good amount in case, you know, we ever
Don't want
wanted
to
to do
mislead
more.
anyone. Yeah, exactly. To be able to preserve it for future study as well.
And it's frozen right now in like a -80 Celsius, I think, freezer. So yeah.
Yeah. Do you know, this is way off topic and I'm sorry. I mean, it's not entirely off topic, but do you know if any of the seeds from these critically endangered ash are kept in the International Seed Bank or in heritage collections and resources like that? Or is that way you'd have to beg off for that answer?
Yeah, I'm not actually 100% positive, but I think I remember reading, I think Cornell might be New York's. So I think there's one university, I think in every state that does this type of job. I think it's Cornell, but I don't know for sure. So yeah.
And then as you've gotten into this area with new species, have you found yourself working with different collaborators that you wouldn't have worked with previously? Do you want to say a little bit about the other lab that you've been working with?
Yeah, so well, the other lab that I work with at UB, it's almost like we're a joint lab a little bit, but the work that I've done in my lab also spans all over the world. So some of the carnivorous plants we work with, those actually were collected in Australia, and we have collaborators there. On one of the first slides, I showed Tembusu, the $5 tree. That's done with collaborators like in Singapore, in Europe as well. So yeah, we don't really work with too many native plants, so we find ourselves working with kind of people from all over the place. So which is fun to have that type of communication with with people that you probably otherwise wouldn't really be having a professional relationship with.
For sure, yeah. So we are officially just past time. I'll say that it brings us to the official end of our program. Thank you so much to everyone for joining us today. Thank you very much, Steven Fleck, for sharing the research that you've been working with. You can stay up to date with Conversations in Science by looking on our website, which is sciencebuff.org. And then our next Conversations in Science will be astronomy at the museum. So from the seeds to the stars, we're going to think about all the ways that people can get engaged in astronomy and education at the Museum of Science. And you can always view past Conversations in Science recordings on our YouTube channel. So you can just take a search for that. So thank you again for everyone for joining us today. Thank you so much to Steven Fleck. Thank you to the Albert Lab, and have a good evening.